High-throughput sequencing has now revealed that our oceans and soils are teeming with a massive diversity of bacteriophages. This chart tracks phage research since 1960. There is a stark divergence between the blue triangles, representing newly sequenced phage genomes, and the red squares, tracking functional publications. The exponential growth in sequencing provides a vast library of raw genetic code, but the physical context remains elusive. A sequence alone cannot explain how a virus actually behaves inside a host. Bridging this gap requires methodologies capable of analyzing viral mechanics at scale. Editors Martha Clokie, Andrew Krapinski, and Rob Levine have compiled the laboratory protocols required to link these sequences to their biological functions. This research aims to translate raw data into application-driven biotechnology, including new diagnostics and targeted phage therapies. While genomic data provides the necessary code, the physical mechanics of the infection cycle are only confirmed when they are physically mapped and validated in the lab. Genomic data alone does not reveal when or where specific viral genes activate inside a bacterial host. To solve this tracking problem, researchers use phagefish. The process begins with dual probe design. One set of RNA oligonucleotides identifies the host bacterium. Simultaneously, multiple double-stranded DNA probes target the phage. These microscopic interactions are difficult to see without significant signal amplification. This amplification is achieved through catalyzed reporter deposition. Horseradish peroxidase antibodies bind to the phage probes and catalyze the covalent binding of fluorochrome-labeled tyramides to cellular proteins, exponentially increasing the visual signal. Dual-color microscopy then provides a clear readout. As seen in Panel F, researchers can visually differentiate between early infection, advanced infection, and active lysis. By connecting sequences to these visual states, phagefish creates a dynamic map of the infection timeline. Beyond tracking the location of the virus, researchers must understand how it redirects the host's metabolism through protein-protein interactions. This diagram illustrates the affinity purification workflow. The process begins by tagging a selection target and initiating a phage infection. Key proteins from a pathogen act as bait to pull down interacting viral proteins onto a purification column. The complex is then separated on an SDS phage gel and analyzed via mass spectrometry to identify every protein involved. This workflow relies on a proteogenomic approach to analyze the data. By searching results against all potential reading frames, researchers discover active phage genes that standard databases often overlook. Mapping these protein networks reveals the specific molecular interactions phages use to hijack their hosts, providing direct targets for novel antibacterial research. To complete the infection cycle, the phage must break open the host cell using endolysins and virion-associated peptidoglycan hydrolysis. The challenge is identifying which specific protein in a complex cell extract possesses this hydrolytic activity. To find it, researchers use zymography. A standard SDS phage gel on the left is run alongside a zymogram gel on the right, which is embedded with an opaque layer of susceptible bacterial cells. Both gels use SDS to denature the sample, forcing the proteins to migrate downward based purely on their molecular mass. After electrophoresis, the zymogram gel is washed to remove the SDS. Without the denaturing effects of the SDS, the separated lytic proteins fold back into their active conformations. The renatured enzyme then digests the bacterial substrate surrounding it, leaving a distinct clearing zone in the opaque gel. By linking molecular mass directly to enzymatic action, zymography identifies active antimicrobial proteins from raw mixtures without the need for prior purification. Identifying these natural functions allows for the engineering of specialized biotechnology. One application involves constructing bacteriophage-based biosensors to detect specific pathogenic bacterial strains. This requires modifying the host range of the virus and inserting a reporter system, such as luciferase genes, into the phage genome. To isolate successful recombinants from wild-type phages, engineers use the enrichment workflow shown in this schematic. Recombinant lysates are plated across decreasing concentrations, forcing them to compete against wild-type strains. Researchers then select the exact wells exhibiting the brightest bioluminescent signal at the lowest phage concentration. This systematic selection transforms a natural virus into a sensitive diagnostic biosensor. Before engineered phages can be deployed, they must prove their efficacy inside a living host. Standard mammalian trials are slow and expensive, making them difficult to use for high-throughput screening. Invertebrate models, such as the Drosophila melanogaster in these flasks, provide a parallelable system for testing systemic pharmacokinetic properties. Fruit flies and a silkworm larva shown here are utilized to model systemic pathogenesis for threats like Pseudomonas aeruginosa and Staphylococcus aureus. Researchers also use avian models, such as the chicken embryo lethality assay. The avian model is highly relevant. Its amniotic fluid mimics vertebrate defense mechanisms, allowing researchers to accurately screen for virulent isolates. These alternative models provide a scalable bridge required to move laboratory discoveries toward human therapeutics. As the library of known sequences continues to grow, the focus of applied virology is shifting toward functional, physical translation. We are now mapping spatial architecture with phagefish, isolating molecular mechanisms via protein purification and zymography, and validating efficacy through high-throughput models. This integrated methodology allows researchers to extract actionable data from raw sequences. Applying these rigorous engineering protocols to the planet's most abundant biological entities is establishing the bedrock for the next generation of precision diagnostics and antimicrobials.